In this video 3 for Control of Gene Expression for Neuroscience lecture, we will keep talking about techniques to measure transcription. Particularly, we will talk about in situ hybridization, microarrays, and next generation depth sequencing or RNA seq. In situ hybridization refers to the use of radiofluorescent or antigen-labeled nucleic acid oligonucleotides. This can be DNA or RNA, and they are called probes. These are used to bind to and localize a target RNA within tissues or cells. Probes are either cDNAs of the target RNA or a complementary RNA, which is called riboprobe. It consists of forming an artificial double-strand nucleic acid, cDNA mRNA or RNA mRNA in situ. And this quantification requires microscopy image analysis. This technique is mostly used to identify and localize a gene transcript, but it can also be used to quantify levels of that particular transcript. In this slide, you can find a diagram of how RNA in situ hybridization procedures occurs. We will start with, for example, a frozen mouse brain that you can slice into cryosections and mount on a glass slide. Then these samples will suffer different pre-treatment steps prior to the hybridization step. One is fixation, that is necessary to fix the tissue and immobilize the RNA molecules where they are. And also a permeabilization step, so then you allow the probes to access to where the transcripts are. After these pre-treatments, we follow the hybridization step, in which the tissue is put in contact with the labeled probes. And basically, this is what is happening. We have the mRNA messenger that will be hybridized by the labeled probe. And as we mentioned earlier, there are different ways of labeling probes. Depending on which labeled probe you decided to use, you may need an amplification step, for which is required an RNA scope that is necessary to amplify the signal of the labeled probe binding to the messenger RNA. Then after this hybridization and amplification step, we will follow with different washes and a dehydration step. And then again, depending on which type of labeled probe you use for the RNA in situ hybridization, you will require different image techniques. One is autoradiography if you are using radio-labeled probes, or confocal imaging if you're using fluorescent probes. This is an example on how to apply RNA in situ hybridization. In this case, there are two genes of interest that we want to know how they're distributed, where they are localized. So in this case, I just name gene A and gene B, and then you generate probes that specifically target each of these two RNAs with different fluorophores. For gene A, we will be using a red fluorophore. For gene B, we will be using a green fluorophore. So then you can detect the expression of those two genes simultaneously on a brain section, and then using confocal microscopy imaging. And then you can see how these two different genes distribute differently, are expressed differently across the entire brain. And then there are different profiles in different regions on where these genes are expressed. And if using confocal microscopy imaging, you can also detect different fluorescent intensity levels that can be interpreted as different amounts of RNA, and then use that value to quantify the expression. This is a different example in which the authors of this paper applied radio-labeled probes to detect different RNAs. In this study, they used a battery of different genes in the expression and analyzing the expression of these genes across the cerebral cortex between human and mouse. And you can see in these images how different genes distribute differently across the different layers of the cortex. It's important to say that by using radio-labeled probes, you can only test one gene at a time. So you're limited by how many genes you can probe at the same time, compared to immunofluorescent labeled probes that can be combined together in up to 4 or 5 different fluorophores. Now, talking about the advantages and disadvantages of using RNA in situ hybridization. Well, some of the cons of this technique is that you're limited by the number of target genes you can analyze simultaneously. In the case of radio-labeled probes, you will be analyzing just one gene at a time. In the case of fluorescent probes, you can use four of the 5 different genes at a time, but certainly no more than that. So you're really limited by the number of genes that you can measure. It's also highly sensitive to degradation, and the presence of RNases can certainly degrade the sample and make your experiment fail. And it's not user friendly. In the case of radioactivity, I mean, it's a problem when working with it. You will require a specific facility and specific instrumentation to work with it. In the case of fluorescent labels, you will also need confocal imaging. You will require, in some cases, an RNA scope, etc. So it's not very user friendly. As I just said, it requires image analysis that may not be possible in different labs. On the other side, you can localize the mRNA so you don't lose this tissue or cell context, and then you know exactly where your transcripts are expressed. Plus, it's also high specific and it has a good sensitivity. So another well-used technique to determine transcription are microarrays, or also known as DNA chip. This refers to the use of complementary DNA or oligonucleotides generated from at least 2 different libraries or conditions or samples to compare the relative expression of a large number of genes across samples. So probes are either single-strand cDNAs or oligonucleotides. And with the use of microarray, we can identify and quantify gene expression changes from a large number of genes. And that's a big difference compared to qRT-PCR that we described in video number 2, in which you can only determine a series of number of genes. In this case, we're talking about hundreds of genes that can be analyzed simultaneously. Here there is a diagram of how microarray procedures it happens. As I just said, you will need to compare 2 different samples, in this case control and a treated sample or tissue or cells. And then you continue with mRNA extraction from those 2 different samples and cDNA synthesis. Then what we do is that the 2 different cDNA syntheses will be fluorescently labeled with 2 different fluorophores. For this particular example, we labeled the control sample with a red fluorophore and the treated sample with a green fluorophore. So now what we do is we combine those 2 samples together into the same tube. We mix them both, and then we apply that mix to a DNA microarray. This is a tiny piece of array that contains several hundreds of genes that have in every single of these wells there are oligonucleotides that will specifically recognize one cDNA. So for every well, we'll represent one different gene. And then when we mix these two samples together and apply them to this microarray, then there will be fluorescent signal depending on whether that specific transcript gene is expressed in one sample or another. So in the case of the green spots, it means that in the treated sample, the expression of those genes is higher than in the control sample. In the red spots here, this means that that particular transcript is highly expressed in the control sample compared to the treated. And then all the yellows are transcripts that are equally expressed in both conditions. Then we will need to scan this array and do comparative analysis of all the different spots that are differentially labeled with one color or another. And then there is a data analysis that will tell you the amount of genes that are upregulated or downregulated compared to the control conditions, and to what extent or fold change there is that regulation. Well, some of the disadvantages for using microarrays is that you always need confirmation by using an alternative technique. Most of the hits or some of the hits that you will find using microarrays will need to be validated by using RT-qPCR, since the use of large microarrays can give you false positives or false negatives. There's also heavy data mining. You're analyzing hundreds and hundreds of genes from different samples, and this requires a specific data analysis and specific knowledge to be able to compile all that data. It also relies upon existing knowledge about the genome sequence, and certainly if you're working with an organism that you don't know the genomic sequence, then you can't apply microarrays. And then comparing expression levels across different experiments it's often difficult and can require complicated normalization methods. As I mentioned, you'll be working with two samples at a time and looking at the relative expression between those two samples. And if you have a lot of different samples to analyze, you'll require multiple experiments and multiple normalization methods to be able to analyze all that data together. On the other side, there is a large number of genes you can test at a time, and this is a big advantage compared to the other techniques for measuring transcription that we've seen. Plus, it has good specificity and good sensitivity. And chips are also commercially available, so then you can buy them in bulk, and they can certainly be very inexpensive. Plus, it's a quick technique, and very shortly you can get hundreds of genes and the expression levels of all of those genes. So now the next technique is the next generation or deep sequencing, or very commonly known RNA seq. RNA sequencing refers to the use of high throughput approaches to study gene expression patterns. Specifically, RNA seq enables genome-wide assessments of gene expression levels, but also including introns and interspersed repeated sequences, transposable elements that are previously considered as junk DNA in other techniques, in other procedures, and they're really important to understand transcription regulation. So, what is the big advantage of RNA seq? Well, about 88% of the genetic variants, or single nucleotide polymorphisms, or SNPs, are currently associated to complex diseases and traits by genome-wide association studies rely within intronic or intergenic regions. This evidence strongly suggests these nucleotide variations are likely to have causal effects by influencing gene expression rather than affecting protein function. So with RNA sequencing analysis, you can get to pick all these SNPs that you wouldn't see when looking at total protein levels, for instance. So RNA sequencing is providing a big advantage in how we understand transcription regulation. In this slide, you can see the different steps required for RNA sequencing. There are three major steps that are necessary. One is library preparation, the second one is step sequencing, and the third one is data analysis. For the library preparation, we start with purifying messenger RNAs using oligo-DTs bound to beads, as we described in video number 1. Then, once we purified all the poly-A messenger RNAs, we proceed to fragment the RNAs into small pieces and prime them with small random hexamers that will bind randomly to every single piece of RNA. Then the next step is to synthesize the first strand cDNA. And then we will follow with a second strand cDNA synthesis. Then all these fragments suffer different treatments to adenylate the 3' sides and repair the 5' ends. And then we will follow with the ligation of different adapters that will bind to every single double-strand cDNA. All these adapters have a barcode that will allow the system to identify which adapters are bound to which transcripts. And then by the use of universal primers that bind to these adapters, there will be a ligation step and a PCR amplification. From these adapters, we will amplify this fragmented RNA, and then we will do the sequencing of every of these fragments. And then for the data analysis, we will do an alignment against a reference transcript or de novo assembly, where all these different contexts, these different amplification products will be assembled to a reference transcript to determine the entire sequence of the RNA. Now, pros and cons of using RNA sequencing. Well, some of the cons is that the use of short or mid-sized reads lowers coverage and depth of the analysis, and this is certainly something that Illumina and other platforms are working on to improve this problem of having really small, short or mid-sized fragments. It can also be expensive, and again, I think this is something that some universities are working on to make it more available to researchers and decrease the prices of this analysis. But it needs a sophisticated normalization of data sets, and there is a really large amount of data that it needs to be aligned and analyzed, and you will probably need a biostatistician or a specialized person to analyze all this data. On the other side of the pros is that you will have the whole transcriptome analyzed, and you can identify splicing, editing, SNPs, all different changes into the RNA molecules. Plus, you can also get quantity information, so you don't only get a transcriptome analysis, but also you get quantity of all these transcripts, and it's high sensitive and high specific. Here there is a short quiz that you could do at home after watching this video, which there are 3 different questions about the use of the different techniques for different purposes, and we will see these questions in class, and some of them will also make it into the final quiz.